For this second portion of membranes, we'll talk more about the different structures that we see within membranes and some of the ways that channels and pumps can be used. But first, let's discuss membrane fluidity. The membrane, as we've discussed, is composed of phospholipids, and those phospholipids have fatty acid tails, and they can either be saturated or unsaturated. And the saturated fatty acid tails pack really tightly together because they're straight chains of hydrocarbons. This reduces the fluidity as well as the flexibility and permeability of the membrane, and that's for saturated fatty acid tails. On the other hand, unsaturated fatty acid tails have one or more kinks in the tail due to the double bonds, and those double bonds in moving or reducing the amount of hydrogens can be cis or trans in orientation where the hydrogens are at. Because they are unsaturated, they pack more loosely, as we see in the picture here, and that makes the membrane more flexible and permeable. And so membranes overall contain a mixture of both of these. It's not one or the other. It's both types of tails, and the ratio really depends on the temperatures that the cells typically experience. Colder oftentimes is going to be more unsaturated, and warmer temperatures is going to be more saturated. Another molecule that's found within the membrane that helps to change the fluidity and permeability is cholesterol. And cholesterol is a type of steroid. It's mostly hydrophobic, but one end of it has a hydroxyl group, which is hydrophilic, and so it's able to fit within kind of both portions of the membrane, but it doesn't extend into the extracellular matrix. It's positioned between different phospholipids within the membrane, as we can see in the picture here, and it typically makes up about 20 to 40% of the membrane itself in eukaryotic cells. The phospholipids are packed densely together, but they're able to move laterally. Cholesterol helps to maintain the arrangement of the lipids, the phospholipids, and helps to stabilize the membrane, particularly at higher temperatures. They also help to ensure that the fatty acids do not solidify at low temperatures, become solids. And so the cholesterol helps to maintain the structure of the membrane, and as well as depending on the temperature. Many cells contain small structures called vesicles that are used to be able to transport or store materials, sometimes water, sometimes waste, sometimes food. And these structures, the vesicles, separates the cell environment from what's ever inside of that environment, and they do so by having a membrane. And conveniently, it's the same. It's composed of the same thing as the cell membrane itself. So it's a phospholipid bilayer, and it's essentially a spherical sac, has liquid or other particles inside. It's commonly found in eukaryotes. And the vesicles have a continuous cycle of creation and destruction or absorption because of the fluidity of the membrane and its shape. And so the creation of vesicles is sometimes to move contents or whatever is inside of them to embed within the membrane, or sometimes they're also used to send or receive particles from in or outside of the membrane. And so the making of these vesicles comes from a small section of the membrane is pulled in from the rest of the membrane and pinched off. And so then it is essentially pulling a part of the membrane and pinching it off so it becomes its own capsule, spherical sac. And this process is carried out by proteins in the membrane, and it does require the use of ATP. So it would be a form of active transport. There's two ways that this can occur depending on whether materials are moving in or out of the cell. The first is endocytosis, and endo being in and cytosis referring to the cell, so it's moving something inside of the cell. And if a membrane is created and contains material from outside of the cell, maybe water or solutes or other particles that are unable to move across the membrane, if it's bringing it into the cell, that would represent endocytosis. And a good example of this is found with a fetus, a developing baby, and a mother. The mother's blood proteins are passed to the fetus. Amoeba and paramecium are organisms that consume prey using this method, and it would be the pinching off of the membrane to bring those materials inside of the cell. The opposite of this is exocytosis, so something exiting the cell, and this occurs when the vesicle fuses with the membrane to transport and then release contents out of the membrane. Can be used to expel waste products, such as like expelling excess water. Can also be used to release necessary proteins, hormones, for example, that are not waste products. And so if it's something that's released that's a waste product, it's called excretion, would be the release of a waste product. Secretion would be the release of a necessary molecule, like a protein, for example, digestive enzymes and hormones. Those would be the representation of secretion. And so both of these, endo and exocytosis, change the size of the plasma membrane by either adding or subtracting, because the vesicle is either being pinched off or fusing with the membrane itself. The phospholipids are synthesized at the endoplasmic reticulum, and then they get moved to the rough endoplasmic reticulum membrane. And ribosomes is what make the endoplasmic reticulum rough, the presence of them. And so those ribosomes that are on the ER synthesize proteins that then get embedded into the membrane. And then this membrane that just has those recently synthesized proteins pinches off to form a vesicle, and that vesicle then fuses with the plasma membrane. And when it does that, it increases the surface area by a small amount. So this is how the membrane actually expands and grows. And this same process we see happen in organelles like mitochondria that have their own membranes as well. The membrane is made up not just of phospholipids, but also proteins, as we've discussed previously. And these proteins can serve as channels, and in this case, specifically gated ion channels. And they allow specific ions to move across the membrane in either direction. And so this would be like potassium ions or sodium ions or other ions, something that's positively, negatively charged. It happens through facilitated diffusion because there's some protein that is enabling the ions to be able to move. And those proteins, in being facilitating this movement, can open or close or essentially turn on or off at specific times. And so the channels themselves must be specific. For example, sodium channels only allow sodium, but not the larger potassium ions to be able to pass across. And so potassium dissolves in water, and the water molecules themselves form a shell around the potassium. And so this increases the size of the individual potassium ion because it's got water attached to it. And so it's not able to move through this ion channel that's designed for sodium. In order to be able to actually move, it has to have those water molecules broken off, and then it can form some bonds with amino acids to move through a narrow part of a pore, which is another protein, and to be able to gain passage into the membrane. And so essentially the controlling of ions moving across the membrane occurs through gates, channel proteins, facilitated diffusion that can be turned on or off and are specific to the unique ions. A more detailed example that we can examine this is the use of nerve impulses using a voltage-gated sodium-potassium channels. The nerve impulses use rapid movements of sodium and potassium ions to be able to move across the membrane. And the channels are made of 4 subunits and a globular protein subunit. Kind of looks like a channel with a chain and a ball attached to that chain. And it can be opened or closed with the ball and the chain used as a way to temporarily block the channel. So there's an on and it's off, it's open, it's closed, and there's also a temporary closing or blocking of the channel, which is a reversible change. That conformation shape can be changed back to be able to be opened or closed. And so how this works is that there's voltages are created due to an imbalance of positive and negative charges. If there's a negative charge, then there's more positive ions present to potassium outside of the cell. And so if the voltage is below 50 millivolts, negative 50 millivolts, which is a measure of electric potential, the channel remains closed. And it remains closed until if the voltage is above negative 50, the sodium channels open up and it allows sodium ions to diffuse in, those sodium ions being positive. Once the voltage reaches positive 40 millivolts, potassium channels open up, allowing those potassium ions to fuse out of the neuron. And so it's a series of gates for each of these that are opening or closing based off of the electrical charges that then controls when sodium or potassium can come in or out of the cell. To send these nerve impulses, there must be an establishment of the concentration gradients of sodium and potassium. And how these gradients are established is by the use of active transport using what's called the sodium-potassium pump. And so this is different than what we've just discussed with the ion channels. This is a pump, and because it's active transport, energy is going to be required. And this is an example of an exchange transporter, as it's transporting different ions in opposite directions across the membrane. And so it uses ATP to be able to facilitate this changing shape of the membrane protein, and it helps to establish charge imbalances and membrane potential in order for ions to be able to move across those membranes. The process of using the sodium-potassium pump begins when 3 different sodium ions bind to this pump, 3 sodium ions that are inside of the cell, and ATP is used, and as it provides energy, it releases a phosphate, becomes ADP, and that's use of the energy, presence of the energy, then causes a conformational change, shape change of the membrane protein, and it then opens up to the outside of the cell, thereby releasing the sodium ions to the outside of the cell. At that point, then 2 potassium ions bind to that same membrane protein, and when binding then are released, the protein itself changes shape again and opens to the inside of the cell, thereby allowing the potassium ions to come inside of the cell. The use then of the sodium-potassium pump and the individual sodium and potassium gated ion channels allow nerves to be able to send signals, and when you touch or feel something, that's how the chemical signals are being sent throughout our nervous system. At this point, we've now looked at different channels that are used to move ions. We've discussed pumps to be able to pump things against the concentration gradient. It's also possible for some molecules to indirectly be transported against the concentration gradient as another molecule moves down the concentration gradient. And a good example of this is the sodium glucose co-transporter in kidney that's used to be able to reabsorb filtered glucose from the blood. And so this protein, this co-transporter protein, transfers both sodium and glucose molecules across the membrane into the cell. The absorption of glucose depends on sodium ions being greater outside of the cell, and the concentration gradient is established by the active transport of sodium out of the cell using the sodium-potassium pump. And so that uses energy, so it's active transporter, but the co-transporter itself of sodium glucose does not, so it's an indirect use of active transport, and it's a byproduct of that sodium concentration being established. As we see in the image, as sodium and potassium pumps are moving sodium ions outside of the cell, that establishes a high concentration, really high concentration of sodium ions outside of the cell. And as we know, molecules want to move from high concentration to low concentration. And so the only way that they are able to do that is through this co-transporter protein. And so we can see as sodium moves through, this protein then also pulls glucose, which has a hard time being able to move across the membrane on its own. It pulls glucose with it and thereby brings glucose inside of the cell along with the sodium, thereby acting as a co-transporter, indirect form of active transport. In order for organisms to have tissues, cells of the same type must be joined together. And so the adhesion of cells to be able to form tissues is made possible by a number of different junctions between cells. And so cells are linked by these different types of junctions, and it depends on the cell adhesion molecules, or CAMs. And CAMs are typically proteins with portions of them embedded into the membrane and others extending out into the extracellular environment, so outside of the cell. And the junction is the junction between these two cells is formed by CAMs in adjacent cells binding with one another and binding together. If the same type of cells are, if the same type of CAMs are present, they bind together to form tissue. If they're different types of CAMs, then it links different cell types and can link those two different cell types. What this does is it forms and maintains tissues and organs and the needed relationships between different adjacent types of cells. And so that concludes our examination of membranes and how things move across the membranes. In our next and future videos, we'll look at how cells become specialized to carry out different types of functions.